So it's my pleasure to introduce our first speaker for this session today. Dr. Amy Petry is a nutritional physiologist and assistant professor in the Animal Science Department at Texas Tech University. Dr. Petry earned her BS and MS at Texas Tech before moving to Iowa, where she earned her PhD at Iowa State University with Dr. John Patience. Her dissertation research focused on investigating the modes of action of xylanase in pigs fed corn-based diets, or corn-based fiber. At Texas Tech University, Dr. Petry is building a multidisciplinary swine research program to investigate three main themes, including the physiological and non-nutritive functions of bioactive compounds of ingredients, improving the utilization and energetic contribution of complex carbohydrates, and the link between inflammatory stressors and metabolic disruptions on whole body energetics. In addition to her many research accomplishments, Dr. Petry has also received several awards throughout her graduate and professional career, including the Young Scholar Award and the Staley P.O. Outstanding Swine Nutrition Graduate Student Award from the Midwest ASAS section. So please join me in welcoming Dr. Amy Petry.
Thank you, Lee. And good afternoon, everyone. Thank you to the program committee for the opportunity to talk on xylanase, which Lee alluded to was the focus of my PhD work here at Iowa State University under John. And my goal for this presentation today is to leave you all with a better understanding on the mechanism of action of xylanase in pigs-fed corn-based fiber, and how we can translate that mechanism of action into successful applications for improving pig health and performance, as well as a few research considerations moving forward when we're evaluating other carbohydrates. So as I'm sure many of us can appreciate, meeting the energy specification of the diet is a daunting and expensive task. And when we think back to John's talk this morning about the different components in the diet that provide energy, protein, starch, fat, and fiber, we see some interesting responses when we increase our utilization of corn co-products. And what I want to draw your attention to, and which will be pivotal to understanding some of the mechanisms today, is that when we increase the utilization of DDGs, there's no question that we increase the amount of fiber in that diet. But the contribution of fiber to net energy is relatively insignificant to the pig, less than 5% of the net energy value of that diet, even at a level of 20% neutral detergent fiber. And so John's lab and myself, we were asking the question, how can we improve the energetic contribution of fiber in the pig and capitalize on this challenge, but also an opportunity? And so one classification of technologies that have been available to producers for quite some time are carbohydrates. And we think back to that talk on fiber earlier about hemicelluloses. The main hemicellulose that you can find in corn and corn co-products is arabinoxylan. It's this complex hemicellulose composed of pentose sugars, xylose and arabinose, substituted with a variety of polyphenolic compounds and substitutions in lignin. And I have a very simplified graph of it here, but I want you to picture a three-dimensional complex structure that spans both the primary and secondary cell wall of the plant that is actually a mechanism of the plant to use for defense against pests as it builds more lignin. And so it's a really complex structure that we're looking at breaking down. And one potential enzyme that can help hydrolyze this complex structure is xylanase. Xylonase is a carbohydrate that targets these beta-1,4 glycosidic bonds, releasing a variety of different components that can either be metabolized if it's monosaccharides such as xylose or pentose, or fermented in either oligosaccharides or polysaccharide forms. And we know about from the poultry industry that this enzyme is pretty effective in attenuating those anti-nutritive effects that are commonly associated with higher fiber diets. But when we evaluate the efficacy of xylanase in corn-based diets within swine over the past 20 years, we see some considerable amount of variability. in that only 33% of the time, or 26% of the time, was an observation in growth performance observed. Yet over half the time, we saw an improvement in apparent ileal or apparent total tract digestibility of fiber. And we classified fiber as observations of either total dietary fiber, non-starch polysaccharides, or neutral detergent fiber. And we looked at the literature that solely evaluated diets that contained cereals that were either corn or corn co-product based. At the same time, we're seeing this considerable amount of variability and this disassociation between digestibility and growth performance responses. We're seeing some interesting things during this era of gut health, gut function work in the feed additive space over the past 5 to 7 years, where there's been a considerable amount of preliminary evidence to suggest xylanase can improve markers of gut barrier integrity, function, structure, architecture, and improves and alters the immune function of the animal under enteric disease stress, as well as there's some indication it might improve oxidative status. And so collectively, when we were trying to think about and put together my PhD work, there clearly needed to be an understanding of the in vivo mode of action of this enzyme in pigs fed corn-based fiber, so then that we could maximize its efficacy in swine diets. At the same time, what was interestingly being observed in field studies and commercial practice was this phenomenon that xylanase may be potentially improving pig viability, or in other words, reducing finishing pig mortality. And so not always a significant response, but certainly has been repeatable that with xylanase supplementation throughout the finishing phase has reduced finishing pig mortality, both in high health herds and in low health herds, as well as in one production system, D. Boyd alluded that xylanase yielded a net live margin of $2.50 per pig placed under conservative market conditions. And so this begs the question, what are the underlying mechanisms of action for how a carbohydrates enzyme can improve pig viability in the pig? And so this led me to the overarching focus of my dissertation work, which was to elucidate the mechanism of action of xylanase in pigs fed corn-based fiber. And so what were we interested in? We put together 5 hypotheses that were either commonly cited within the literature as potential mechanisms of action of this enzyme, and also evaluated a mode of action that was new and a little bit outside of the box. And so the first mode of action we focused on is pinnacle to the success of this enzyme and all subsequent hypotheses that we evaluated, and that xylanase does indeed, in situ within the pig, digest arabinoxylan to some extent, releasing metabolizable and fermentable components. Modes of action 2 and 3 that we evaluated is how did those released products impact the microbiome? Did they elicit a prebiotic-like effect that selectively upregulates beneficial microbiota that confer a health benefit? Or did they stimulate the microbiota to ferment fiber beyond just the released products of arabinoxylan itself? Modes of action 4 and 5 we investigated evaluated more about this nutrient uplift and digestibility response that's somewhat observed with xylanase. And number 4 evaluated this idea that we can release trapped nutrients or mitigate this nutrient encapsulation effect of fiber and release other nutrients that might otherwise not be available to the pig. And number 5, we evaluated does xylanase attenuate the negative physiochemical properties that are associated with feeding some high fiber diets. And so what I'd like to do today is to dive into some data supporting or disproving some of these hypotheses, and then get into a cohesive mode of action that I would like to propose as a stepping point forward as we move with xylanase in the industry. And so to test these 5 hypotheses, we used 2 different experiments that were similar in dietary treatments, which we'll get into. And I don't want to get into the minutiae of materials and methods, but just want to say that we had considerable amount of adaptation time in the experiment that largely investigated the mechanism of action of xylanase. And we did a metabolism study with these pigs and then necropsied them and took a variety of samples from all over the gut. So small intestine, large intestine, and measured a variety of different gut health markers. And then experiment 2, we used a dual cannulated pig model where we equipped these pigs with an ileal cannula as well as a cannula in the medial jejunum to evaluate the efficacy of this enzyme across the small intestine. And we did this over time. And so we left these pigs on the same dietary treatment for 52 days and separated our collection so we could evaluate the impact of adaptation time on the efficacy of xylanase. And among these two experiments, we had similar dietary treatments where we used a low-fiber dietary control, which would be kind of your typical corn, soybean meal-based diet. We then used a high-fiber control where we added 30% corn bran without solubles, which is a rich source of corn-based arabinoxylans that has not been subjected to microbial fermentation, mostly at the expense of corn. And we allowed the energy within these diets to float and didn't add any fat, so they were not isocaloric, but were isonitrogenous. We then took that same high fiber control and added xylanase at 100 milligrams per kilogram in order to achieve 16,000 Birch-xylane releasing units per kilogram of diet. We then took that same high fiber control and we added 50 milligrams per kilogram of an arabinoxylane oligosaccharide that we thought may be the potential in situ release product of xylanase. And so what did we find? So first, we'll dive into this mode of action, and it follows this logic. We feed these pigs this corn-based arabinoxylane composed of these pentose sugars, substituted with ferulic acid and other polyphenolic compounds. It hydrolyzes these bonds, and from in vitro work, we know it can potentially release xylose, arabinoxylane oligosaccharides, low molecular weight arabinoxylans, or arabinoxylane oligosaccharides that are substituted with ferulic acid. And indeed, what we did observe is that pigs fed xylanase here in this blue bar had an increase in the ileal digestibility of non-starch polysaccharides compared to that of the high-fiber control. And this can be largely attributed to the improvement in arabinoxylane degradation compared to that of all other treatments. And so this is the first indication that we're getting some breakdown of that fiber in the small intestine of these pigs. But what's even more fascinating is what we move into the large intestine is what happens to that fiber that's been broken down. What you're looking at here on the left is the flow of neutral detergent fiber through the pig. So this in the purple is the amount of neutral detergent fiber that disappeared in the small intestine. The blue is the amount that disappeared in the cecum. The green is the amount that disappeared in the large intestine post the cecum, and everything in orange is what was excreted in the feces. And what I want to draw your attention to is if you compare this bar here, we see a nearly threefold increase in the amount of fiber that disappeared or potentially was fermented in the cecum of pigs fed xylanase compared to that of that high fiber control. And this was in addition to an improvement in total short-chain fatty acid production. But in addition to that, which was interesting from a microbiome perspective, which I'll get into a little further later, is that we had a shift in the molar proportion of short-chain fatty acids and that the microbiome favored acetate production over butyrate and propionate. So we've seen what happened in the small intestine and large intestine, and there's certainly some indications that we're getting fiber degradation and fermentation. But does this translate into an energy response? Well, we did see a decrease in the amount of energy that was excreted in the feces. But what was interesting is that we did see an uptick in the amount of energy that was excreted in the urine of pigs fed xylanase. And this may be indicative of xylose metabolism, as work from my colleague Nicole Huntley suggests is that as pigs metabolize xylose, they excrete more xylose as well as xylose metabolites from its metabolism in the liver. So it gives us some indication that there's likely some pentose metabolism going on in the pig because of this increased urinary GE output. However, although we did see an increase in urinary excretion of energy, we did not affect the efficacy of this enzyme to improve the metabolizable energy value of the diet and an estimated net energy value. So indeed, in this situation, it does appear that xylanase is improving the energetic contribution of fiber. But did this translate into a growth performance response? The objective of this study was not to evaluate growth performance. We weighed these gilts to understand what was going on, and we did see an improvement in average daily gain. But what I find even more interesting is the amount of time it took for average daily gain to be improved. As you can see, it took between 14 and 27 days to see an improvement in average daily gain in pigs fed xylanase. And it was even greater after between days 27 and 36. And we saw a similar response for feed efficiency. And when we followed this up with that dual cannulated pig trial, we saw a really interesting response in that upper small intestine, so from the all the way into that medial jejunum, and that with increasing adaptation time, the apparent jejunal digestibility of insoluble dietary fiber with the supplementation of xylanase increases. And this is probably the first that we're aware of that this upper small intestine adaptation effect has been studied, and certainly gives us some indication that we have to give this enzyme its time to work and elicit its mode of action in order to degrade this fiber, and may translate into those later growth performance responses. And we saw a similar response for total dietary fiber, neutral detergent fiber, dry matter, and gross energy, and recently followed this up and saw an improvement in arabinoxylan degradation as well. But what was released? That has been the big question for us, is we know the fiber is being degraded, but what is released by the enzyme? It's not a really easy question to answer because the methods in order to evaluate oligosaccharides within the ileal digester are highly complex. But within collaboration with Dr. Gansel at the University of Alberta, we evaluated the oligosaccharide pattern of ileal digesta of pigs fed xylanase. And we gathered some preliminary evidence that suggests that indeed xylose, xylobiose, as well as oligosaccharides that likely because of their elution time, although because standards are not yet being created, we can't determine what their composition is, have a degree of polymerization between 3 and 7. And so there's certainly likely some oligosaccharides that are being released from this enzyme within the small intestine of the pig. And so what are those oligosaccharides doing in the gastrointestinal tract? And that's what mode of action 2 and 3 that we evaluated really dealt with. And so it's this concept that is it eliciting a prebiotic or a symbiotic-like effect. And I think when we're evaluating these things, we need to understand what a prebiotic is and what a symbiotic is. A prebiotic has been most recently defined as a substrate that is selectively utilized by host microorganisms, conferring a health benefit. So there's 2 parts. Has to be selectively utilized by microorganisms, must confer a health benefit. A symbiotic, on the other hand, is an additive that stimulates a fiber-degrading microbiome, resulting in an increase in fiber fermentability, even though the additive itself contributes little to short-chain fatty acid production. And so to evaluate these 2 modes of action, we analyzed a variety of different GI function and physiology markers, as well as we evaluated the 16S rRNA sequencing of the microbiome communities in 6 different gastrointestinal locations in these pigs, as well as we characterized their predictive metagenomic function for being able to degrade arabinoxylans and produce microbial carbohydrates, metabolize pentose sugars, and produce short-chain fatty acids. And from this project, we gathered a considerable amount of data. And I don't want to get into the minutia of what OTUs were upregulated and all of the details to the microbiome, but would rather summarize 2 key publications as it relates to the prebiotic and symbiotic effect of xylanase. When we feed pigs xylanase in a high corn fiber diet relative to that of a control, in the small intestine, it appears we're eliciting this prebiotic-like effect, where we're differentially upregulating 3 microbial communities that are known to establish cooperative microbial metabolism of arabinoxylan oligosaccharides: Lactobacillus, Bifidobacterium, and Fecalbacterium. And it's been well established in the human sciences that these microbial communities, when working together, increase butyrate production as well as gut health. And indeed, we did see an increase in short-chain fatty acid interconversion, where they were taking acetate and producing butyrate from this microbial metabolism, and we'll discuss it a little bit in more detail later on. Additionally, upregulating microbiota is only one part of the prebiotic definition. We have to confer a health benefit. And what we observed in the small intestine of these pigs is an upregulation in the mRNA abundance of collagen and gluten, which are tight junction proteins that span the paracellular space. As well as we saw an improvement in total antioxidant capacity of these pigs, which might be indicative of ferulic acid uptake from the polyphenolic structure of the corn-based fiber, as well as a reduction in the amount of malondialdehyde, circulating malondialdehyde, which is a marker of oxidative stress. However, when we transition into the large intestine, we see a different mechanism of action being upregulated by this enzyme as it relates to the microbiome. and that we're still upregulating lactobacillus and bifidobacterial microbial communities, but we see an increase in microbial diversity. And this is largely driven by arabinoxylin degrading microbiota, Lactobacillus, Rhamnococciaceae, and Prevotella that are known to produce their own microbial carbohydrates and further degrade that corn-based fiber outside of just xylanase alone. And this may partially explain the improvements in fiber digestibility and short-chain fatty acid production that we observed in the cecum of these pigs that I presented earlier. Additionally, when we look at the metagenomic function of the microbiome, we see a considerable amount of increase in the microbial carbohydrates, upregulated related to arabinoxylin degradation, as well as pentose metabolism. So collectively, it appears this enzyme is doing just more than breaking down fiber. It's eliciting a gut health response by working collaboratively with the microbiome. But what remains of that fiber structure after the enzymatic reaction? And this is this concept that we can release trapped nutrients. And so to put it in perspective for you, we fed these pigs bran, which is this whole and fiber portion of the corn. And if we zoom in on that bran, during dry milling, they take off the bran between the aileron and endosperm layer. And in the cell walls of this layer, it's connected by arabinoxylans. And so our hypothesis is that if this enzyme is breaking down arabinoxylans, it's pitting the cell wall and breaking open that structure, potentially releasing some of these nutrients that were otherwise inaccessible to the pig. And this has been commonly cited in the literature as a potential mechanism of action because we often see with xylanase supplementation and other types of fiber an improvement in nutrient digestibility outside of fiber and dry matter, such as phosphorus, calcium, nitrogen, and crude protein. However, we wanted to look at it from a little bit of a different perspective. And so we used scanning electron microscopy imaging to evaluate corn bran in the ileal digesta of these pigs. And so what you're looking at here, on the far left is a picture of scanning electron microscopy picture of corn bran that we fed these pigs. And the middle is the ileal digesta from pigs fed that high fiber control. And on the far right is that high fiber control with the addition of xylanase. These images were taken and selected by a research technician who was blinded to treatment and to the hypothesis to prevent confirmation bias, and was asked to select a picture that represented the image, represented the group of images. And although subjective in nature and not objective, we can see that there's some pitting of this fiber structure and a greater amount of surface area with xylanase supplementation. And when we zoom in 500 times, it's really interesting to see the amount of fiber degradation and breakdown of that corn bran in the ileum of pigs supplemented with xylanase. The fifth mode of action that we evaluated did not deal with really what the fiber structure, but the influence of fiber on the physicochemical properties of digesta. And so we evaluated the digestive viscosity of the ileum, cecal, and colonic digesta from these pigs. And we did it creating a shear rate curve, so to evaluate the non-Newtonian function of the digesta. And what that really means is we just measured the digesta at multiple shear rates and then took the intercept of this line as the digestive viscosity value. And what we found is that xylanase did reduce the digestive viscosity in the ileum, but where xylanase's efficacy appears to be the greatest, there was no impact on digestive viscosity. And we're of the opinion that it's likely not the case that xylanase is mitigating a viscosity effect in corn-based diets, as corn-based fiber is highly insoluble and non-viscous in nature and doesn't create the viscous gels that are associated with more viscous fiber types. So collectively, this is a large project with a lot of data. When we put it to paper, what is the mechanism of action of xylanase? And so to set this slide up for you, over here in the blue, this is what's going on in the lumen of the pig. This is the epithelial layer, and anything down here is what's happening systemically. We fed a pig corn-based arabinoxylans supplemented with xylanase, and our hypothesis is that it would release a variety of different products: xylose, arabinoxylin oligosaccharides, low molecular weight arabinoxylans, or these arabinoxylin oligosaccharides that are substituted with ferulic acid. and indeed did confirm that the arabinoxylane structure was broken down in the ileum by the improvement in the apparent ileal digestibility of arabinoxylane, as well as there was likely some released products that had a degree of polymerization between 3 and 7. We know that xylose can be absorbed through the epithelial layer through an unknown mechanism transporter system. And it can contribute to energy in the pig through the pentose phosphate pathway when the mechanism is not overabsorbed from a high level of xylose. It then can contribute to pyruvate and ultimately ATP, as well as it can be excreted by the pig as xylose itself or one of its many metabolites. And these may partially explain the improvements in average daily gain, feed efficiency, and metabolizable energy that was observed, as well as the increase in urinary GE output. And it appears from both our work and Nicole Huntley's that this increases with increasing adaptation time. And when we think about the symbiotic mode of action, or what's happening in the large intestine of these pigs, it appears that these released products of the enzyme are upregulating microbial communities that produce their own carbohydrates. And a lot of these accessory enzymes that are then further breaking down this fiber and improving its fermentation. And this is increased with the amount of time, as well as it increases the amount of fermentation of the fiber and short-chain fatty acid concentration, because now we have a microbiome that can produce its own carbohydrates and favors the fermentation of arabinoxylanes. And when we move into the prebiotic-like effect, it's really this cooperative microbial metabolism that may partially explain the pig liveability that we've been observing in these field trials. And so the arabinoxylane oligosaccharides that are released from Xylonase are then first degraded by arabinoxylane degrading microbiota such as Ruminococcacea or Lactospiraceae, as well as lactobacillus. The lactate produced from lactobacillus is consumed by lactate-consuming bacteria, as well as lactobacillus produces shorter arabinoxylane oligosaccharides, ferulic acid oligosaccharides, as well as acetate. We know that Bifidobacterium preferentially metabolizes shorter arabinoxylane oligosaccharides as well as xylose oligosaccharides and produces acetate. And interestingly enough, fecal bacterium is a poor utilizer of carbohydrates. It has no metagenomic machinery for digesting carbohydrates or sugars, but facilitates on acetate proliferation, particularly in the mucosa, and colonizes next to Bifidobacterium and produces butyrate. As well as we know that Bifidobacterium contains ferulic acid esterase and has the potential to release ferulic acid or ferulic acid arabinose complexes, which are potent antioxidants from the arabinoxylane structure. And so this was partially, this kind of cooperative microbial metabolism that I'm proposing was partially supported by our improvements in acetate and short-chain fatty acid, butyrate production, as well as increase in metagenomic potential for pentose metabolism and short-chain fatty acid interconversion. And so we know that acetate and butyrate can be absorbed by the pig through sodium-dependent monocarboxylate transporters, as well as ferulic acid can be passively diffused through the epithelial bilayer. Acetate and butyrate are thought of as energy sources or short-chain fatty acids. They can contribute to fat synthesis. Butyrate is the known energy source for colonocytes. But I'd like for you to expand your thinking on short-chain fatty acids outside of just their potential to provide energy, but also as potent modulators of gut health and immunity. As we know that acetate can influence the NLR3P inflammasome pathway of the innate immune system, reducing inflammation within the pig. As well as butyrate can modulate transcription factors inhibiting histidine acetylase and thus opening chromatin and allowing more transcription of pathways that will upregulate gut health. As well as ferulic acid is a potent antioxidant that is locked within the cell the minute that it enters and can mitigate reactive oxygen species. And collectively, these mechanisms may partially explain the improvements in average daily gain, feed efficiency, Claudin-4 and occludin upregulation, as well as the improvement in total antioxidant capacity that we observed, and the systemic and localized improvements in total antioxidant capacity and malondialdehyde. Now, I don't want to make a profound conclusion on just 2 projects and just a small data set, but I certainly think there's some evidence here for us as an industry to lean on these enzymes and other enzymes for improving the health and viability of pigs, as well as some future research avenues as we move forward with enzymes. And so collectively, when I think about my time spending in the literature with xylanase and other carbohydrates, what are some thoughts on successful strategies for using of carbohydrates and how we can implement this mechanism of action within the pig? First, I think it's really important as nutritionists that we understand how carbohydrates fit into our formulation system and with our ingredients and their expected responses. The mechanism of action of xylanase that I presented here today is likely different from that of wheat or in the presence of rye, barley. There's likely some similar overlaps, but we know that there are different physiochemical properties and different fiber structures found in those cereal grains. And so it's not a one-size-fit-all situation when it comes to enzymes, and we have to think about them selectively with the fiber that we're providing in our diets. We also have to think about the expected response. We know that xylanase tends to have greater efficacy in wheat-based diets, and from a performance side, it hasn't been the most effective with DDGs, but there's certainly some evidence to suggest that it can work with corn-based fiber, and so it's figuring out how we can elicit that mode of action, and adaptation time might be part of it. We also have to think about targeting the right enzyme with the right substrate at the optimal dose. There's a multitude of different carbohydrates available on the market, carbohydrates products that have multiple different enzymes with them produced from a variety of different sources. And of my opinion, that we still don't have a good understanding of what microorganisms produce certain enzymes and how their efficacy with certain cereal types. And so does the Bacillus subtilis-based xylanase work better with wheat versus corn? There's some preliminary evidence by Nadeau et al. That suggests those things. There's certainly more research that is needed. Also, when we think about the fiber types in our diet, if we're going to use carbohydrates, we need to target enzymes that fit those fiber types. And so in corn-based diets, it's arabinoxylan and potentially cellulose. And where other diets, it might be some mannans, glucans, or if you're getting in the sugar beet pulp section, maybe pectins. And so we have to think about the fiber types that are there. We also need to think about an optimal dose and how that optimal dose was founded. Certainly there's some production systems that start feeding enzyme as early as the 25-pound mark, but they don't feed it at the level that they feed finishing pigs, and they titrate it greater into the finishing side. And so we need to think about what is the optimal dose in your production system, particularly when we want to feed it for a longer amount of time to give it ample adaptation time. And particularly, how was that optimal dose created? It's been pretty evident in the literature that in vitro work with enzymes does not often translate into in vivo work. And so when you're thinking about what dose you're putting into the enzyme, ask your supplier of that enzyme, how did they come up with that dose? Did they use a growth trial? Or are you using your own growth trial in your system to determine what the optimal dose of the enzyme should be? I'll reiterate this point for probably the 10th time, but I think we have to give the enzyme ample time to elicit its mode of action. And it's my suggestion that at least 25 days in corn-based fiber diets with xylanase. And it's due to this upper intestinal adaptation that we still need more research to understand what's going on there from a mechanism standpoint, but there certainly seems to be something happening with an improvement in fiber digestibility. And so when we think about this mode of action and this time response, and we're giving it ample amount of time, we also need to think about how that diet changes over time. And so if we're constantly changing our formulation, from one phase or one least cost formulation section to the next, it's likely changing the fiber types that are there. It's also important as an industry we consider how we value the carbohydrates in our system. There's no question that growth, performance, average daily gain, feed conversion are powerful KPIs in production. However, I think there's a case to be said when we consider the mortality responses with xylanase that it is also a powerful KPI that can increase your return on investment when supplementing an enzyme. And so consider how you're evaluating an enzyme in your system when you're going to try a new one or supplement an enzyme. And certainly, in order to understand these KPIs, we have to be recording them. My 5th recommendation is this adage is that the diet you formulated is different from the one the manufactured, different from the one delivered to the feeder, and likely different from the one the pig consumed. And so if you're anticipating an expected response with an enzyme and you get a different response, maybe consider testing that enzyme activity of feed batches when variable responses occur. And maybe we need to be testing them at the mixer or also at the pig level to understand, is that enzyme activity truly there? Finally, it's important that we consider the health status of our herd because the pig has to consume this enzyme for it to elicit its mode of action. And so when we have a phenotype where pigs pull off feed, such as PRRS, it may not have ample time in order to elicit its mode of action. However, there's some evidence to suggest there might be a preventative effect under certain disease stresses if we're supplementing the enzyme early enough to affect the microbiome. And so there's this kind of catch-22 that if we can supplement the enzyme long enough and establish this cooperative metabolism of in the microbiome, there might be a preventative effect for disease if it hits the pig later on. So what are some research considerations with xylanase and other dietary carbohydrates? The first one I keep going back to is, should we be targeting the depolymerization of the entire fiber structure, or should we be targeting products for optimal fermentation and host microbiome symbiosis? And so is our objective to break down that fiber into monosaccharides, so either pentose monomers if we're thinking about arabinoxylans, or glucose when we're thinking about cellulose? Or is the better payoff with the enzyme fermentation? Can we target the enzyme for optimal host microbiome symbiosis and elicit these types of health effects that I have proposed with this presentation? Also, with enzymes, it's pretty evident in the literature that in vitro studies do not consistently translate into in vivo findings. We often see in vitro studies that show an improvement in fiber digestibility, but yet maybe don't see that in an in vivo setting. And so we need more comprehensive, multidisciplinary system-based research to understand how these enzymes are working and where they're best applied in production. We also need more comprehensive reporting of study designs. And so we need to think about, as researchers, when we're reporting our studies, what are the valuable information that somebody else would need when doing this research in the future? And some things are, we need to be reporting the enzyme activity and testing it, the pig's mortality, the chemical composition of the diet, appropriate fiber analysis. So if we're using a diet high in soluble fiber, we might need to be considered total dietary fiber analysis. And so certainly within the literature, we need to consider how we're reporting study designs and be more holistic in that approach. We also need to consider what are the limitations of using carbohydrates in the presence of other feed additives. There's certainly no question that we're not just using one feed additive within a production system. It's likely multiple. And do they have competitive or complementary mechanisms of action? Are we canceling out the mechanism of action of an enzyme or potentially another feed additive, such as a DFM, when we're supplementing them together? Also, in research, we need to consider the ubiquitous use of certain feed additives, such as phytase, in our research design. This research was done in the presence of phytase because we know that the majority of pigs fed are fed phytase. And so when we're thinking about the research of the future, we need to be thinking about those feed additives that are going to be consistently fed to pigs as their economical choices for producers. And kind of outside of the box, can we design doses and combinations of enzymes to tailor fermentation to the energetic needs of a specific physiological process? So thinking back to Peter Thiel's data from this morning, can we tailor the energetic needs of farrowing? Can we increase beneficial short-chain fatty acid production to help improve that farrowing response? Can we improve maybe disease through certain combination and tailored designs of carbohydrates packages? And with that, I'd like to thank the multitude of collaborators that were a part of this project and USDA and NIFA for funding my PhD work, and we'll open the floor for questions and discussion.
Thank you, Dr. Petry, for that talk. We'll go ahead and get the question section started now.
Yes, Amy, this is Raphael. First of all, Very nice presentation. Excellent understanding of xylanase. We have done some work in the past looking at xylanase increasing the digestibility of fat. And the reasoning there is non-starch polysaccharides, if left intact, they're going to increase the hydrolysis of bile salt or bile acid, which in fact we need them for the digestion of fat. I would like to see if you can add a little bit more understanding to that effect, if you have done some similar type of work.
We measured the digestibility of fat in both of those studies, and we didn't see a difference with supplementation of xylanase. Sarah Weiland's work did see some interesting response as it relates to fat digestibility with xylanase. Certainly the concept that you put forth with NSP, breaking down bile salts and that physiochemical property in the small intestine is there, but I think it's somewhat dependent on the amount of fiber that is present and the types of fiber. So if you're really insoluble, certainly that can happen. Also, if we think about soluble fiber types and some more of those soluble arabinoxylan sources, they can actually prevent the digestion of fat by preventing it from lipases, so kind of creating this barrier within the small intestine and preventing that emulsion. And so that might be one way that we're improving fat digestibility, is that we're breaking up some of that bilayer and helping the emulsion of fat. But it's kind of a hard response to explain on the fat digestibility side. I don't think it's pertinent from my studies on corn-based fiber.
Yeah, I really appreciated your presentation, and I know your research was done with one of the xylanase sources. You know, obviously there's several sources on the market, and a couple parts of it, would any in vitro trials be helpful in terms of helping to understand the differences between sources? And then also tied to that is, can you ever use too much of a xylanase in a diet?
For your latter question, yes, I think there's certainly some evidence that we can use too much xylanase, particularly when we're thinking about what production stage of pigs. So there's that mortality response tends to be at an optimal dose, and so if we get too much, it increases back up. And so this is likely because we might be releasing too much xylose, since we kind of get this negative effect on the gut. And so there certainly seems to be, it has to be at an optimal dose. And it's hard to say what an optimal dose is, because if we look at the variety of different xylose products on the market, the units in which we define xylanase are different. You know, everybody kind of has their own version of defining what xylanase activity is. And so of the different types of xylanase on the market, I certainly think there's a role for in vitro work to define maybe early on, does it degrade certain fiber types better than others. But we've done, that work has been done, particularly in the Netherlands, and some work in Bach-Knudsen's lab, and then they put it into an in vivo setting, and it doesn't translate at all. And so I think we're at the point in the industry that we've got to take the step forward and go into those in vivo trials if we're really going to understand and get the payoff of understanding what microorganisms produce xylanases that might fit better with certain cereal grains.
Really nice job with this. One question I have, and I like how you set up the right enzyme, right substrate, right dose. How do we sort through that, though? Because I agree that's what's needed. And as we get into different diets, I mean, obviously we go to distillers, but wheat midds, corn germ, you know, the different byproducts we bring in that have the different fiber types, and then the right type of enzyme to go after that, or wheat-based diets. You know, how do we begin to sort through to be able to answer that sequence that you talked about?
Yeah, it's a really great point, and I think we have to think about it from a couple different perspectives of what is our objective of using the enzyme. So if we're wanting to maximize energy, maybe it's the one that has the greatest amount of substrate. And so maybe we're using multiple carbohydrates if their mechanism of action aren't overlapping. I think it's a bit hard sometimes to say which situation is better, but from my perspective, I think you've got to look at the gamut of ingredients you have, the different fiber types, and say, What is the most abundant substrate that I can target? What gives me the most chance to break something down? And that's what I would probably do from my perspective. And so when we think about corn germ and DDGs and corn-based products, it's likely arabinoxylans. When you get into wheat, you've got some other things. Soybean meal certainly has its own fiber types that we have to consider, and particularly when we get in those soluble fiber sources. I think there's an era coming where we can probably advance into more multi-carbohydrates use, but it has to be tailored. I don't think throwing a multi-carbohydrates in there at a gambit and expecting it to solve all our problems is the right answer either. We've got to be more strategic in how we approach those substrates.
I'm getting there.
I appreciate your comment about multi-carbohydrates. Could you comment on that concept a little bit more? Because my understanding is most of what we call xylonases in the market today are not just xylonase. They're multiple carbohydrate enzymes, so maybe they should just be called NSPases, even though we measure xylonase, and that's what would be on a COA, because that's the one that we can measure easily. We should all be aware that they pretty much all contain multiple carbohydrates, not just xylonase.
Yeah, I think you're right on that point. You know, these single vat fermentation systems, we've got multiple different carbohydrates coming out. But I think we have to give some credit to a lot of R&D teams that are working on single enzyme application, that they've teased out the genetics of those microorganisms to really upregulate the expression of producing one enzyme. And so yes, there's some accessory enzymes there, but not maybe at a level that's really going to have a magnitude of effect. Certainly, I would say, I agree with you. When we think about xylonase, it's likely not just xylonase, but it's mostly xylonase compared to some enzyme cocktails on the market or multi-carbohydrates products where we're putting everything that was fermented into the product. So I think it's both there, but certainly there's some enzymes on the product that are the majority is just that single enzyme product. But we have to keep it in the back of our mind that there's likely still just trace amounts of other enzymes in there as well.
I think we have time for one more question. But if not,
I'll ask.
one more.
Thanks. Amy, the other question that I had was, there is a nice publication from a Chinese lab. Actually, they look at, specifically during the nursery, when they were feeding a xylanase, there was, and you showed some of that work, that shows an increase in beneficial bacteria and also a decrease in pathogenic bacteria, E. coli, Salmonella. You showed increase in beneficial lactobacillus, bifidobacteria. I would like to get your opinion on that.
Yeah, I think I know which paper you're talking about. They saw a differential upregulation in Lactobacillus reuteri and an E. coli challenge. Yeah, there's certainly some evidence with the arabinoxylin oligosaccharide work done in human nutrition that microbial triad of Lactobacillus, Bifidobacterium, and Fecal bacterium, they thrive on establishing this cooperative metabolism on those types of oligosaccharides. It provides the perfect ecosystem for them to benefit their growth based off of the byproducts of what each one of them metabolize. And Lactobacillus reuteri from that paper is one that breaks down those arabinoxylin oligosaccharides that are about that medium length that might be what we're seeing and released in situ by this enzyme. So I certainly think there's some preliminary evidence there from this work, and I know of at least 3 other papers that have seen an upregulation, if not 1 or 2, of those microbial communities. So I think it's an avenue that we have to think about moving forward.
Okay, let's thank Amy again.